Okay, so what were we doing? So we were doing diffusion. Right? So diffusion. So we wrote down del p del t equal to d del 2p del x2, right? So we wrote down the diffusion equation. This is true in any dimension, and depending on what dimension you have, you get your appropriate mean square displacement, which is 2d d, right, to the power of 1. And you can, because I think somebody was asking, one can think about how to get these exponents other than 1. So that is one of the assignment problems. So you can do that and see if you get some exponent other than t to the power of 1 or not. Then we did this for polymers. We said that this was true for trajectories in space. This was true for trajectories in time, as well as for trajectories in space. And therefore, not only is it applicable for normal trajectories, but for polymers. And from there, we did the ideal polymer. We did these ideal polymers. And we said that this ideal polymer, or what we also called as the freely jointed chain, that acts as a sort of spring, in that purely it acts as an entropic spring. So it acts as an entropic spring. So if you calculate the entropy, the maximum number of microstates is when the polymer is collapsed, and therefore if you are to stretch the polymer, it always will try to collapse. It will try to collapse back. And in fact, that behavior, that restoring force is like k times the displacement, with some spring constant k polymer as something kt by n a square. I think 3 kt. I think 3 kt. Yeah. It's in 3 dimensions. I think it's 3 kt by na square. Okay. So that's what we had done. And then we are talking about these applications. So we had started with this Drosophila. I'll go back to this, but I just wanted to maybe talk a little bit before I move on. I forgot to do that. Is to talk a little bit about the diffusion equation and recasting the diffusion equation a little. So one of the things is, of course, that the diffusion equation is essentially dissipative. Which means that, if you are to start with some rho of x like this, something, if you have a concentration profile or a density profile or a probability density, whatever, something like this as a function of x, and this evolves in time according to the diffusion equation. If you waited long enough, you would get something homogeneous, right? Because diffusion tends to sort of smooth out any hills or valleys. So if you have something like this or something like this, if you waited long enough, so this is diffusion in the absence of any potential of pure diffusion governed by this diffusion equation. In the long time limit, you would get some uniform profile. Okay? So long time limit. And that is easy enough to see in that if you are at the sort of if you are at the top of a hill, right? So this is your rho of x. If you are at the top of a hill, then over here your del 2 rho del x2 is less than 0, right? Because this is a maxima, and therefore your del rho del t at that time, how it evolves in time at a point which is a maxima, is negative, right? So del rho del t is negative, right? Which means that whatever was the local concentration, that sort of tends to come down with time. Similarly, if you are at some sort of a minima, your del 2 rho del x2 is positive, and therefore your del rho del t is positive. So here, if you are in some position like this, it sort of tends to increase the local concentration profile. Right? So that's in that sense, diffusion tries to homogenize everything. It doesn't like hills, it doesn't like valleys. if you are moving purely by diffusion, you want to ultimately reach something which is a steady, like a uniform profile. The other thing is that you can recast this diffusion equation by using continuity, or conservation. Right. Well, and what this means is that if I have a particle which is undergoing a random walk, right? So I have a particle which is undergoing some sort of a random walk. As long as in the absence of any sources or sinks, so let's say it's a single particle or whatever, n particles, but that n is not changing, it's some n particles which are undergoing random walks in some domain, you know, you cannot create or destroy particles, and in that sense your density which describes this random walk, so rho of x comma t, this is locally conserved. And locally conserved in the sense that you cannot have a particle that came here and then suddenly reappeared back over there, right? It has to follow some sort of a continuous trajectory to over there. So if you had something like that, that a particle was here and the next moment it appeared there, it would still be conserved, but it would not be locally conserved. So diffusion and so densities like this are locally conserved in that you have a continuous path. Okay. All right. So you can then recast this in terms of currents. So I have this rho of xt, which is so this is the density of some conserved quantity. Okay, it can be the density of particles, it can be some probability density, whatever. It is the density of some conserved quantity. So this is the density of some conserved quantity, right? And you can also define a current J of xt. All right. So this is the amount of this conserved quantity, per unit area per unit time. Okay. So this measures how is this conserved quantity flowing through space. So it's a spatial current. And then if this conservation equation you can write mathematically as something like this. So it is d dt of integral rho dv, right, plus so this is a volume integral, plus you have a surface integral j dot ds, right, and this is equal to whatever sources or sinks that you have, sum of sources or sinks, right. So I have some density which is changing with time. So I have some volume that I consider V, and inside this volume I have some rho of x comma t that is changing with time. Okay, the only way it can change with time is if things are flowing into or out of the surface, which is why you have the j dot ds over here, or if you have localized sources or sinks inside this volume, which either absorb particles or it creates particles. So in that sense, this change in volume, this change in this quantity must be equal to sources or sinks. If you have no sources or sinks, then this is 0. So this is no sources. Okay. And then if you use the divergence theorem, you can just write this as a volume integral. So this gives you j dot ds becomes divergence of j dv. So what this gives you is del rho, this gives you a continuity, del rho del t plus divergence of j equal to 0. This I think the continuity all of you are familiar with, right? So this gives me my continuity equation. And what that means is that if I talk about diffusion, I can identify a diffusive current because I can recast, I can look at this equation and I can look at that equation, and I can read off essentially what is my diffusion current. So, J diffusion, it is what? So, it is, let us say in 1D, this is 1D, it is d del p del x minus sign, right. So, this is the diffusive current. This is called Fick's law. This is called Fick's law. Not in 1D actually. This is in this. I should not say 1D. Yeah, it's 1D. Or you can just, I could have just written d del p essentially. The subtlety comes sometimes when you have diffusion coefficients which are themselves spatially varying. So, there is no, I have talked about a homogeneous diffusion coefficient which is has this value d everywhere, but one can generalize this to more complex cases, including when if the diffusion coefficient itself is a function of time, or for example, we are talking about the drift diffusion equation. When you have a velocity, if p was not equal to q, you had some sort of a velocity term, and then your diffusion current or drift diffusion current, whatever J, would be V times P minus d del P del x in the case of drift plus diffusion. Okay. So, often people will refer to Fick's law, and you have to then interpret that as the diffusion equation plus continuity to read off what are the diffusive currents. Huh? J is a vector. Here, that is why I wrote in 1D. It is just divergence of J. Huh? So, this fact that in diffusion, you can identify a current, and this current is proportional to the gradient of the density and to the diffusion coefficient. This statement is called Fick's law. That this current due to diffusion is given by this expression. This is called Fick's law. Okay. Anyone else? What is a vector? The velocity? Yeah. So, it's also a vector. Again, I've written in 1D, but if I write in how this term, how does this term come? So, that is that the question? So, that is a part of the assignment, that how do you get this drift diffusion equation? That is, I think, problem 1 of the assignment. But essentially it comes when you do not have symmetric random walks. If you want to move more in one direction as compared to the other, you will get some resultant velocity. Here? Correct. So, V is what? V is delta x space over time. Then here also I have, remember, delta x square by time, that was my diffusion coefficient, and 1 over 1 delta x is sitting over here. So, this is also delta x by time. So, dimensionally it sort of matches up, right, the both of the terms of this. Do it and see, do it and check. All right. So, what we were talking about is this Drosophila, to get back to, just to give you an example of why, where 1D diffusion plays a role in a biological context. And if you remember, we had this Drosophila embryo, right, and there were these nuclei which after some time migrated to the surface of this embryo. Right. So, these were the nuclei. inside this nuclei are your DNA and so on. And then the bulk of over here is this yolk, and in this region in the cortex there is this cytoplasm. Okay, so this is a yolk, something like this. And we were asking, how do patterns get established in a situation like this? And we talked about this chemical bicoid, and the RNA corresponding to that chemical is deposited at one pole, and that pole becomes the anterior pole. And then you have synthesis of bicoid. So bicoid is produced from here. So the bicoid protein is produced from here. It diffuses and it also degrades, and then it produces some sort of a pattern. So a few things to note. One is that these are the nuclei, and you'll see that every nuclear division, the number of nuclei sort of double. So let me play this again from the beginning. So initially there are very few nuclei, and then when the nuclei divides, it becomes double in number, it divides again, and it becomes double in number, and so on. And when this bicoid protein is produced, it flows only through this area of the cytoplasm. It does not get into the yolk. Okay. So although you might think that there is a protein which is being produced and which is spreading, it is not purely three-dimensional diffusion. It is not three-dimensional diffusion because this yolk region is inaccessible to the protein. So it's diffusing only through this cortical region. So this is some protein which has been tagged with a green fluorescent protein, so that when you see this green intensity, that is this protein. So it's being produced here, and slowly it will spread out. But you'll see it will never spread out into this bulk. It is spreading only along these regions, if you see with time. Okay. So these proteins are sort of produced and they spread out only along this cortical layer. And therefore, if I think about modeling these, often an effective one-dimensional sort of diffusion equation is enough to capture the gradients of these proteins. So again, you see that the highest concentration is where the protein is being produced, and then slowly it spreads more and more in that direction. Is this setup sort of clear? All right. So how do people go about modeling this? So the simple thing is to write down something like a diffusion equation, right? So you produce the bicoid protein. The bicoid protein diffuses, so it has some diffusion coefficient d, and it also has some lifetime. So it's a decay process, which means that it has some characteristic lifetime tau. So it's like a Poisson process, and then together the time evolution of the bicoid protein is governed by these two processes. It diffuses and it degrades. So this is called as the synthesis. The fact that there is synthesis, so it's called as the synthesis diffusion degradation SDD, the synthesis diffusion degradation model. And the synthesis is essentially encoded in this part. One can write down this boundary condition in different ways, but this just says that it is being synthesized at that x equal to 0 point. So therefore, the maximum concentration is over there. Or you could just say that del C del x at x equal to 0 is some q, which also says that at this point there is some source. So you could use that sort of a boundary condition as well. You'll get the same result at the end of the day. This one, 1 by tau. So it says that I have this protein. Once the protein is produced, it has some lifetime. It has some finite lifetime, after which it degrades into the constituent amino acids. Okay. So the assumption here is that the decay process is Poissonian. So you have some rate r. So with some rate r, it decays at some instant of time, and therefore you will have some. If you look at the lifetime of these proteins, you will have some characteristic lifetime, which is 1 over r tau, right? And therefore, if you think so, if you just had a first order decay, right, nothing else. So del n del t is equal to minus n by tau, something like that, right? This is just like, or minus kn essentially. So, this is just that equation, tau is inverse k. So, the symmetry is broken by the fact that the RNA is deposited at one point. So, wherever that RNA is deposited becomes the head site. So, this RNA deposition, which is the source, so this is my source, wherever that has been deposited, that is your x equal to 0 point, because that breaks the symmetry, and then the protein spreads out from the source. Now, you could ask that, given this sort of an equation, what would be the steady state profile, the steady state concentration profile of this bicoid protein? And the steady state concentration profile is, just setting the time derivative to 0. So, if I write some concentration of bicoid to 0 and solve this equation, you will get some sort of a concentration profile for bicoid. What will you get? So, if I want to solve this equation where the left hand side was 0 and I ask what is the concentration profile of bicoid as a function of x, what would I get? Exponential, okay, exponential of what? Minus 1 over d tau, root d tau, and times x, and similarly according to the boundary conditions. Very good. So, if I take this as a large length, so if I take this as, I can take a semi-infinite length, so it's 0 to x less than 0 to L, and then if I take a L tending to infinity limit, only the minus x terms will survive. But you can take a finite length also, it does not matter. Like you said, you will have a sum over 2 exponentials, with one with a minus x and one with a plus x. But essentially, if my system is long enough, if my system is large enough, I will get this sort of a profile e to the power of minus x over lambda. So, you will get some sort of a characteristic length scale, right, and this characteristic length scale is going to depend on your diffusion coefficient and the lifetime of the bicoid protein. So, you get some e to the power of minus x by lambda, where this lambda is square root d tau. So, therefore, the bicoid protein, if I think about this Drosophila embryo, there will be more bicoid protein here, less here, and so on and so forth, with a profile that looks like this, e to the power of minus x over lambda, and this lambda depends on the diffusion coefficient and the time scale and the characteristic time scale. Before I talk about this figure, let me just say that does this match up? So, if you look at this, if you look at the profile of bicoid in experiments, you can measure that, because again you can tag these with fluorescent proteins, measure the fluorescent intensity, and thereby characterize the profile. So, if you do that, experimentally you find length scales. So, this lambda value, this lambda is roughly of the order of 100 microns. I forgot to say this embryo is of the order of roughly around 500 microns for the one species, Drosophila melanogaster. So, it turns out that this length scale lambda is of the order of 100 microns, and you can check whether the numbers sort of match up. So, one can measure diffusion coefficients of proteins or whatever other molecules inside the cell. In fact, hopefully I'll talk a little bit about that today. So, if you measure the diffusion coefficient, that's of the order of micron square per second, something like this. You can measure the lifetime of the bicoid protein, which is something like that. And then if you put it in this root over d tau formula, you get a length scale which is of the order of 120 microns. Huh? Where are we considering the curvature? Huh? We are not considering the curvature, at least in this model. But technically one should, because this is a curved, it's it's some sort of a ellipsoidal geometry roughly, if you look at the Drosophila embryo. So, you can do that. You can solve the diffusion equation on a curved geometry and find out what would be the length scale. it turns out that the correction is not really very important. So, the major and the minor axis of the Drosophila embryo are such that the correction factor that you get is not too important. So, you can still do an effective one-dimensional approximation. But yeah, in principle, one should do it on a curved surface, and people have done it, of course. People have done it on a curved surface. Okay, so that's nice. So you do this simple one-dimensional diffusion with degradation. You work out the steady state, and you get some sort of a length scale, which depends on the diffusion coefficient and the time scale of the characteristic time scale or the lifetime. You go back and put in the numbers, and it seems that you get things which are roughly sort of what you would expect. So, and of course, I give these one number 100 microns or 3000 seconds. Of course, experimentally there is a lot of variability. So the fact that you are getting roughly same numbers means that you are roughly on the right track. Okay. Yes. Correct. Yeah. Is that the question? So people actually, so once you have these fluorescent proteins, you can actually go and measure these concentration profiles as a function of x, but also as a function of time, and check that whether ultimately after some time they keep changing or not. So this process, this establishment of the Bicoid gradient happens roughly over a time scale, I would say, of around 2 hours. If I remember, it's roughly around 2 hours. So people have actually done systematic measurements in time to check whether it reaches a steady state, and it seems like for the Bicoid at least it does. That after some time, again, I thought I had it, but apparently I don't. So yeah, there are nice experimental curves which say that the profile does not change with time. So therefore, you can use a steady state profile. Okay. Well, yeah, I mean, there are conflicting views also, but some people, there are theories which say that one should take the time dependent thing. It's just that, but anyway, mostly I think people agree that the Bicoid is roughly in a steady state. So these are like these intensity profiles as a function of position along the embryo. One thing is that if you do this sort of a theory, what are the shortcomings? So one is that this tells you that the characteristic length scale, provided your system size L is larger than your lambda, this lambda that you calculate, this characteristic length scale should not depend on the size of the embryo. It's just a function of your diffusion coefficient and of the lifetime of the protein. Okay. But then you can do this experiment in different sort of Drosophila species, different fly species, and each fly species has a certain length. So something is around 400, another Melanogaster is around 500, 600. This is a much longer one, Sericata, which is around 1500. And the assumption is that it's the same Bicoid protein that is diffusing. It's the same. It has the same lifetime and the same diffusion coefficient. Therefore, the lambda, the characteristic length scale, should not change. But essentially, what you can see is that very clearly, for example, for this long embryo, there is a scaling of the profile with the embryo length. Right. So the longer the embryo, the larger the lambda, and that is what is shown in this next plot over here. So these are these three species. This is lambda as a function of the embryo length, and it seems that there appears to be a scaling with the embryo length. So it's a nice model to start off with, the synthesis diffusion degradation, because it gives you exponential profiles, which is indeed what you see. In experiments. You do see exponential profiles like this. The numbers seem to sort of match up, at least for Melanogaster, but on the other hand, you don't explain everything, in that you don't explain how these numbers scale with the embryo length, and in that we still don't have a theory for this. It's an open question as to how these the Bicoid gradient length scale, how that scales with embryo size. It's an open question. Right. So like I was saying, so the first step is this establishment of the Bicoid gradient. Okay. So that is my Bicoid gradient over here. It's high at this anterior pole and low at the posterior pole. But in response to this Bicoid, you can have production of other proteins which respond to this Bicoid gradient. And these are some of these downstream genes or proteins: hunchback, giant, Krupel, and you can see they have distinct expression patterns. So, for example, hunchback is more in the first half of the embryo. So these are the profiles, the average profiles, and then a little bit again at the end. This protein giant has these two strips. So one high intensity band over here, another high intensity band over there. and then this scruple is something which is mostly in the middle of the embryo. So, this is scruple over here. Okay. Yes, I mean, it is the characteristic length scale in the sense of an exponential profile. You have an exponential profile, it is described by this length scale lambda. So, it is that length scale, but of course, it does not mean that beyond this lambda there is no protein, because it's just the characteristic length scale in the sense of an exponential characteristic length scale. And then you can sort of build up theories as to, although I am not going to talk about them here, you can build up theories as to how you get, and these boundaries of these proteins are quite sharp actually. So, in that sense, you get these very sharp bands of these proteins, and you can build up how to have these proteins interact such that although the bicoid has this sort of an exponential profile, this is for example hunchback. So, this hunchback has this sharp drop around the 50% of the embryo, and you can show how this hunchback depends sensitively on the bicoid concentration. So, when bicoid is low, hunchback is low. When bicoid is the other way, right? So, you can build up models of binding of proteins depending on this exponential, you now input this exponential profile and then try to predict how these other proteins would behave. Okay. And then you can do this cascade. So, this is called the patterning cascade. Everything starts from bicoid, but then everything responds to that, and you get different patterns that emerge. So, I did not explain because but there are theories. So, it has to do with cooperative binding. So, there's theories which has to do with cooperative binding. Maybe at some point, if I get time, I will talk about that. But essentially, the idea is that you can respond to sort of thresholds in that if this is like I was saying the other day, if you have, so one way to think about is thinking in terms of thresholds. So, if you think as this as some threshold concentration, this is C of x as a function of x for bicoid. If you have some threshold concentration, and if that threshold is a very sharp value, and you express some protein if it's higher than this and some other, then you can get sharp profiles of the downstream proteins in response, although the bicoid is a fairly smooth exponential gradient. But one can make this more quantitative. I'm not getting into this, but there are theories of cooperative binding which gives you these sort of sharp profiles in response to them. So, this is a nice piece of work which shows that although of course there is noise, so this is like the bicoid intensity, and the bicoid intensity profiles have some spread. So, if you were to do it over multiple embryos, you would get some spread, but the thing that responds, this is I think hunchback, thing that responds has a much sort of tighter band. So, although bicoid has some spread, the downstream genes can actually have smaller spreads. So, yeah, again, I'm not going into how this comes about. You can look at these papers, for example, but people have developed theories as to how this downstream sort of patterning cascade occurs as well. So, this is about bicoid. If there are no, I'll just take a stop and then discuss something else. But this was just to give you an example that this sort of one-dimensional diffusion. It's not just a toy model, but you can actually apply even the simple 1D diffusion models to real biological systems in order to understand various real biological phenomenon, in this case gradient formation by bicoid. These are experimental data. So, this is, I think this is from the Shushmanzadeh paper. So, this is an experimental paper. So, people can actually measure all of these proteins intensities as a function of time. Again, you tag it with some fluorescent protein and then so on. All right. So, what I'll move on to talk about next is, so like I said, so if you wanted to find out this length scale for the bicoid, for example, you would have to know what was the diffusion coefficient of bicoid. And so, I'll talk a little bit about how do people go about measuring these diffusion coefficients. Okay. It's nice to know since we are talking about diffusion, how do people measure these diffusion coefficients inside biological systems. Okay. So, one is of course, you could use Stokes-Einstein, get a sense of the size of the protein, and then just do kBt by 6 pi eta r to get some sort of a diffusion coefficient. but that is in some sense an in vitro measurement, because inside cells it's a complex environment. There are nuclei, proteins, everything else. So ideally, you would like to measure the diffusion coefficients inside these sort of environments. There are multiple ways. So I'll just talk about one of the simplest ways to measure the diffusion coefficients. I'll talk about this method, fluorescence recovery after photo bleaching. The idea is very simple. The idea is this, that you have your cell, right? You have your cell and you have whatever protein whose diffusion coefficient you want to measure, right? So you have whatever protein. Okay. These proteins are tagged to a fluorophore, right? Like this green fluorescent protein, which is why, if you were to look at them under a microscope, you see whatever is the fluorescent color. If you tag green fluorescent protein, you observe green intensity. Okay. So all of these, that's what you see in these sort of experiments. Now, in this photo bleaching experiment, what you do is that you select a window and where you have some proteins, and you shine an intense laser light in this window. Okay. What that does is that it exhausts all the fluorophores that were bound to these protein molecules inside this region. Okay. So if you shine an intense laser light, all of the fluorophores that were attached to these fluorescent molecules in this region of interest, they emit whatever photons they had in the excited state, and then they become dark. Okay. So whatever proteins, the proteins still remain, of course. It's just that they are not fluorescing anymore, because you have exhausted out all of their fluorophores, okay, by shining an intense laser light. So then what you will see is that you will have this dark region, and then you will have all this fluorescent intensity outside this region. Now what would happen if you allowed time to pass? These molecules are all diffusing around. So eventually some molecules will come into this region as well. Things will homogenize, because diffusion tends to homogenize. You will get some uniform intensity. Okay. So after some time, and these things which were dark, they will have moved out somewhere else. So this will be dark somewhere else, but because they are spread out, you won't get a concentrated region of darkness. So that's what happens in these experiments. This is a tagged protein. You select a region, you bleach it. When you bleach it, it becomes dark, and then you keep waiting, you keep waiting, you keep waiting. After some time, because these are diffusing, everything else will come back to a uniform intensity. You can measure the intensity in this region, the average intensity, as a function of time, and you can calculate the solutions of the diffusion equation, and therefore ask that how does this intensity recover as a function of time. So I measured this let's say integrated intensity or whatever, average intensity in this window region. I plot this intensity as a function of time. So there was some intensity. You photo bleach it, the intensity drops to 0, and then slowly as things diffuse, the intensity will recover back. This is what you can measure quantitatively from these experiments. How it recovers back, this time scale of recovery, is a function of your diffusion coefficient. And therefore, by measuring this recovery curve experimentally, you can measure what is the diffusion coefficient of these molecules. Is the protocol clear? You are shining a laser on this in this region, so therefore that region becomes dark. It's a circular region. If you have a circular laser, whatever beam of light you shine, that region you photo bleach out. It's not moving the molecule. That's what I'm saying. The molecules which were there, they continue to be there at t equal to 0 when you shine. So let me call this my t equal to 0. The molecules are there. It's just that they are not fluorescing anymore. So therefore, in a microscope, you cannot see them. And then these molecules are diffusing. So whatever one molecule diffuses out, another molecule diffuses in, and so on and so forth. So eventually, you will see a sort of uniform intensity recover back. We are not seeing. You're not seeing a particular protein, but what does this? You're seeing these spots of light, right? So that is because of the proteins are fluorescing. That is what you're observing. So of course, individual proteins are too small, but you have many proteins. Each of them has this fluorescent tag on it, and therefore that is what you're seeing in your microscope. This one, is the intensity versus time or density versus time. Intensity is proportional to the density, of course. Because I write intensity because I'm looking at this in a microscope. So I'm going to capture some light intensity in this region, but that intensity in the diffusion context is just the density, the rho. Yeah, it's just the rho. But when you're doing an experiment, you are seeing some light intensity. Rho is this intent. You can think of this as rho. It doesn't matter. But this the light intensity is just proportional to the density. So I will work out this curve exactly. Okay, so we'll see. Yeah, at least in a simple context. Where? This thing? Huh? I think this is the nucleus. It just has a higher concentration. I'm not sure. I have to go back to the A and look, but this looks like the nucleus of a cell. But you're photobleaching in the cytoplasm. But on the other hand, for example, you could photobleach inside the nucleus and see whether you get a different diffusion coefficient. So people can do different things because the diffusion coefficient can be spatially heterogeneous. So you need not have the same diffusion coefficient. This experiment, this particular one is bleached inside the cytoplasm, but yeah, you can bleach it in different regions. Okay, so the protocol is clear. Yes. So that's what we'll calculate. So that's what we'll calculate using the diffusion equation. Because that's how people analyze. So people solve the diffusion equation. They ask that what will be the form of f, then they match it to the experimental results and read off the diffusion coefficient. Yes? Doubt? No? It's a very simple idea in that sense. And of course, people do more complicated things, FCA, fluorescent correlation spectroscopy and so on. But I like this because it's a nice simple thing and it's still very much used. It's not extremely accurate, but it gives you a very nice sense of the numbers. Right. So this is this long cell. I forget which cell this is, because what I'll do is, again, for the sake of simplicity, I'll solve this 1D diffusion. You can do the same. People have done it for 2D and 3D and so on and so forth. I'll just solve this 1D diffusion equation to obtain this form of f, and it would be applicable in a scenario like this, where you have an elongated cell, and then you have photobleached one region, and then you observe the recovering intensity as a function of time. Okay, so that's the idea. So that's what we'll try to do. So here is my diffusion equation. Let me say, since I'm looking at concentration of molecules, so let me just write C. And what I'm doing, I'm going to consider this FRAP experiment, this fluorescence recovery after photo bleaching, FRAP, in one dimension. Okay. So what does that mean? That means that I have some area. Okay, let us say some domain minus L to L. All right. I had some concentration. I choose a window and I photo bleach that window. So let me call it again just for mathematical simplicity. I choose a region which is symmetric about the origin of length 2a, so minus a to a, and I photo bleach this region. So which means I've shined a laser light on it. The molecules that were in this region are not fluorescing anymore. Which means what? Which means that I have an initial condition. So I have an initial condition which is at t equal to 0, the time when you photo bleach, which is C of x comma 0 is equal to let's say C naught 0 C naught. This is minus L x to minus a x to a, and a to L. Okay. So outside I have some homogeneous concentration. In the photo bleached region, which is a region of length 2a, I have no fluorophores, so my concentration is 0 or my intensity is 0. And again to the right, I have that concentration C naught. Okay. This is my initial setup. And let's say I need some boundary conditions. So let me say that I'll put reflecting boundary conditions. So del C del x at x equal to plus minus L is 0. What I'll try to do is that I will try to expand this concentration profile in terms of a Fourier series. So you can solve it in many ways. So what I have over here is expanding this concentration profile in a Fourier series. Now remember that any function can can be expanded in a Fourier series. So, f of x is sum a naught plus sum over n equal to 1 to infinity a n cos n pi x over L, n pi x over L plus b n sine n pi x over L. Okay, something like this. And these Fourier coefficients can just be read off. So, a naught is 1 over L minus L to L f of x dx. A n is 1 over L minus L to L f of x cos of n pi x by L, and bn's are again 1 by L minus L to L f of x sine. So, any function I can expand in a Fourier series with the coefficients given by these. And what I will try to do is that I will try to expand the concentration profile in this Fourier series. Okay. So, this is my governing equation, my diffusion equation. So, I want to write my C of x comma t in terms of this Fourier series. So, these coefficients become functions of time, of course. So, I will just let me write these as capital. So, a naught of t plus sum over a n of t cos n pi x by L, right. These terms are not there because I have chosen this boundary condition. So, the boundary condition says that the cost terms will be there, the sign terms won't be there, right. So, you can just do del C del x and check that this automatically satisfies the boundary condition. All right. So, I have this series expansion for the concentration profile. I plug it back into my diffusion equation. Okay. So, what do I get? I get del a naught del t plus sum over n equal to 1 to infinity del a n del t cos n pi x by L, right. That is my left hand side, and then I get on the right hand side del 2 del x2. So, this term does not contribute. So, d sum over n equal to 1 to infinity a n of t cos minus n square pi square by L square cos n pi x over L, right. Now, you can solve this order by order for every a n. So, what this tells you is that del a naught del t is 0 and del a n del t is minus d n square pi square by L square a n of t, right, which means you have a n of t is equal to a n of 0 exponential of minus d n square pi square by L square times 5, right. So, solve for the Fourier coefficients, and a naught of t is, of course, a naught of t is a naught of 0. Okay. Now, all that remains is that you need to know what are these initial a n of zeros or a naught of zeros, but that because you know the initial concentration profile, you can just substitute this Cx into these and read off the a naughts and the a n's at the initial time point, right. So, a naught of 0, so a naught of 0 is 1 over L minus L to L Cx of 0. So, this is okay, minus L to L Cx of 0 dx, which is 1 over L minus L to minus a C naught dx plus a to L C naught dx, right. The middle region is of course 0. So, this is L minus A, right. So, this is 2 times C naught into L minus A. Similarly, you can read off what are the AN of zeros. Again, you do this divided by L. Thank you. Similarly, you can do read off these ANs. So, I will just write down the answer, whatever. So, AN, yes. Why is it? Outside the bleached region. So, my bleached region is here. So, this is my cell, I am bleaching this region, this is my window. So, outside I have some intensity, right. No, this I am diffusion is happening all over, right. So, that is what is going to homogenize your density. So, you have a concentration profile here, here you have dropped it to 0 and then you have again some concentration. Then this whole entire concentration is molecules is diffusing, which is why after some time, I mean in the long time limit, you will get back some uniform concentration profile, a little lower than whatever, but you will get back. So, that is why we are solving this diffusion equation in this entire minus L to L domain, because here otherwise how will you do that? So, that is the idea. So, you can write your AN, let me just copy that and write. So, your AN of 0, and you can check this minus 2 C naught by n pi sine n pi A phi. Okay. So, once you have your A naught and you have your ANs, you can just substitute it into this concentration equation to get what is your C x t, C of x comma t. So, that is this C naught 1 minus A by L minus sum over n equal to 1 to infinity 2 sine n pi A by L sine n pi A by L by n pi e to the power of minus d n square pi square d n square pi square by L square into time, and then a cos n pi x by L. Okay. So, you have solved your diffusion equation, your entire concentration profile as a function of time, right. And of course, I have solved it for this very simple geometry, which is 1D. You can solve it for whatever geometry that you have. For simple geometries, you can do it analytically. If it's not a very simple geometry, you do it numerically. But the idea is exactly the same. You specify whatever is the initial condition, which is determined by your laser. What is the shape and size of the laser that you are using to photo bleach? You take that initial condition, you solve the diffusion equation in whatever dimension your cell or your system is in, and then you obtain the concentration profile as a function of time. Okay. Yes. Here, how am I putting? But this A naught 0 is del A naught del t is 0, right. So, the A naught does not evolve. AN part we calculated, right. So, we calculated AN of 0, and then I have put AN of t, which is AN of 0 times exponential of minus dn square, which is why I have this exponential of minus dn square, right. So, this part is AN, this part is the time dependence, and then this part is the cos n pi x by L. Yes, there is a 2 factor where here looks like, right, there is a 2 factor. I do not have it here, Either I've done something wrong here or I've done something wrong here. Huh? There is a 2 factorial. This is a 2L. It's minus L to L. Sorry, I was writing 0 to L. Okay, so Cx of t. Now that is of course the full concentration profile, but you can also do this. Essentially the concentration profile only in the photo bleached region, since that is what people measure. So people just measure the concentration profile in this photo bleached region, and you can call that something. So you can calculate the integrated intensity inside the photo bleached region. So minus A to A C of x comma t dx and call this some NF of t. Okay. So this is the integrated concentration inside the photo bleached region. At the instant that you photo bleach at time t equal to 0, of course here all this concentration is 0 in this minus A to A. So your NF of t starts from there, and slowly as your concentration builds back up, your NF of t will also build back up. So you can do this integration once you because you know the Cx of t, you can just integrate it. You will get some. So let me just write this without doing it, because 2 C naught A, and I may be wrong by factors, but roughly something like this, into 1 minus 1 over A by L, 1 minus A by L into sum over n equal to 1 to infinity 2 by n square pi square sine square n pi by L e to the power of minus d n square pi square by L square times tau. Okay, so you get some complicated whatever expression, not too complicated because my geometry is very simple. But essentially, if you say what is the integrated intensity, it is something which depends on the size of your photo bleached region. So A, the size of your cell L, the background intensity before you photo bleached, okay, C naught, all of which are known, right? Because how much region you are photo bleaching you control, that is your experiment. The cell you can measure the size, the background intensity you can measure, when you photo bleach. So the only thing that is not known here is this diffusion coefficient d. So like I said, you have this curve NF of t, you compare it to the experimental results, and the only unknown parameter is your diffusion coefficient. So you see what value of diffusion coefficient matches the recovery curve from experiments. Okay. Huh? It is just the integrated intensity inside this region, inside your photo bleached region, minus A to A. How is your concentration building back up? Should not change at the edges over here. This is a reflecting boundary condition, right? So what this means, because this means that the current at the edges is 0, because current is d del C del x. So if you have a cell and you have proteins which are diffusing, once it comes to the edge, it has to go back, it has to come back in. So it's a reflecting boundary condition, which is what we have implemented. Depending on if you have some other geometry, of course you can independent, but this is the most standard boundary condition for it, because this says that the current is 0 at the edges. Okay, and indeed, this is actually I don't have an experimental plot here. I have an experimental figure in the assignment, but essentially your recovery curves look like this, and from here you can read out whatever diffusion, whatever is the appropriate diffusion coefficient. The only subtlety, which is what I is also there in the assignment problem, if you look at these actual experimental images, so you would expect that this builds back. If I scale it by the average intensity, it builds back up to 1. So C scaled, it builds back up to 1. Often in experiments, you will see that is not the case. It will build back up to some smaller value, maybe 0.8 or 0.7, which just says that there are a fraction of proteins which are not mobile and a fraction which are mobile, and that has to do with some other biology as well. But that does not change the diffusion coefficient, it just changes the saturation value. So, that is just some other experimental input. It is diffusive in the, no, it is diffusive, the protein is diffusing. This equation has nothing to do with small time scale, this is the diffusion equation, which is why, if you, so this is good to see. If I go back to the images, of course, it depends on, so my cell is roughly of the order of, let us say, 10 microns, right, 10 microns. Diffusion coefficients of these sort of proteins and so on are roughly around 1 to 100 microns square per second. For example, this bicoid, I think I wrote a value which was 5 or 10, I forget, something like that. I think 5 microns square per second was bicoid. But anyway, roughly, if you look at these proteins, it will be in this range, 20, 30, something like that. And the length of your cell, let's say, is roughly around this, okay, which means to sort of scan this whole cell by diffusion will take some time. Actually, I think I have that slide in a different context, but it's good to, because how long does it take to sample a region which is of length x is roughly, so x square goes as dt, right. So, if your length scale is roughly of the order of 1 micron, it takes 0.01 seconds. If your length scale is of the order of 10 microns, it takes around 1 second, and so on. So, to reach this steady state of this sort of a FRAP experiment, you will have to wait of the time scale of some seconds to see this steady state, because your cells are of the order of 10 microns, 30 microns, something like that. That would be one interpretation. Well, what actually happens is that not all protein molecules are mobile. So, essentially, you think of, so here is my region that I am photo bleaching. So, you think of the proteins being into, for whatever reason, you think of some proteins, everything is everything has a tag, but some proteins can move and some proteins cannot move. So, only those proteins will come and diffuse here, which are mobile. These ones which are immobile will actually not stay back. So, it has to do with the fact that a fraction of proteins are immobile, not the fact that it is not recovering. Work it out. So, it has to do with, essentially it has to do with what are you saturating to, how fast you saturate. That is just, that does not, so that value over here is, so if I write it as some A, that changes what A you saturate to, it does not change how fast you approach that saturation. Won't they affect the? In some sense, of course, yes, because this diffusivity that you get is, after all, an effect of the mobility of the individual protein itself, but also of the background in which it moves. And yes, the background contains these other proteins, but they contain so much other things that a few proteins not moving individually, you cannot say will have a major effect. So, essentially, you will get the diffusion coefficient of this protein molecule in vivo inside the cell. So, that's what you will do. Okay. So, before I end, actually I had these numbers just to prove a different point, which is that if you have these cells and you assume a diffusion coefficient whatever on the higher side, 100 microns square per second, you can ask that how long does it take to traverse this whole cell by diffusion, okay. And the time scales are reasonable as long as your cells are of this order, or tens of microns. If your cells become larger than this, then the time scales to explode this full cell by pure diffusion alone becomes very large, because most biological processes have to happen over time scales of seconds. So, if you are having to wait few minutes for a protein to go from one place to another, or I write 1 meter. So, this looks astronomically large, but there are in fact neuronal cells in our body which are meter long. So, this is not an unreasonable number for a cell. Nerve cells are indeed very long, and they can be of the order of meters. And therefore, what I just wanted to say that inside cells, if you are in this range, you can, through diffusion, you can sort of cover the entire cell. However, if you have larger cells, then and you need to transport proteins from one place to another, then simple diffusion will not do. You need some driven process, something which actively moves the protein from one end to another. And again, there is a machinery for the cell to do that. So this is just to show you that simple diffusion would not be enough if you had very large cells. Here are some measurements of the diffusion coefficient obtained through these sort of FRAP experiments. So this is a small ion, which is of course very fast. It's 2000 microns square per second. This is a green fluorescent protein inside the E. coli cytoplasm. Again, so that is the range, 10 microns square per second. And DNA, for example, inside the nucleus is very low, because DNA is this very long polymer. So it essentially does not move a lot. So if you were to look at the diffusion coefficient of DNA, that would turn out to be very low. You can do this for do the same thing for whatever is your protein or molecule of interest and read off what is the diffusion coefficient. Okay, so I think I'll stop here for today.